Hello everyone. In this video series, we'll be covering some important chapters of Claydon. As you know, Claydon is a very good book to start initially with understandings of organic chemistry. So in this video particularly, we'll be studying chapter 15 of Claydon, that is nucleophilic substitution at saturated carbon. So before starting, we should understand some initials. What is nucleophile and what is nucleophilic substitution at saturated carbon? So nucleophile is a species with excess of electron density that is highly electron dense and loves loves positive charge, loves nucleus. So that species is called nucleophile, nucleus loving. Now nucleophilic substitution at a saturated carbon. Saturated carbon means carbon that do not have any pi system. It do not have any pi system, like it is the carbon that has which is particularly sp3 hybridized and has no pi system with it. So we'll be studying nucleophilic substitution at saturated carbon. There are two ways through which this substitution take place: Sn1 or Sn2 substitution. So studying in detail about these two sidewise, and here if I give a brief introduction what is actually given in this chapter. This chapter covers in detail about these two substitutions, how different conditions they favor or inhibit the Sn1 or Sn2, like solvent effect, temperature effect, what are the different neighboring groups that should be present, and all this. So we'll be covering all in some short videos. Here we'll be covering just small introduction about what are Sn2, what are Sn1 mechanized as substitution reactions. So first of all, starting with Sn1 reaction. Sn1 reaction means substitution reaction which is first order in nature. Like if we have any saturated carbon R1, R2, R3, and here is any leaving group. Remember that for substitution to take place, important thing is leaving group. If leaving group is present, then only substitution can take place. A good leaving group is very important. And if this is followed by first order reaction, means that this saturated carbon skeleton system, this undergoes a reaction in which it forms a carbocation and leaving group leaves. In this step, there is no involvement of nucleophile. And intermediate, mind word intermediate, meaning it is the intermediate is the complex in the reaction step that is stable for small time and can be isolated. So carbocation is formed, which further is reacted upon by nucleophile, which is highly electron dense, and to form our product. So here in substitution, Sn1 substitution of first order reactions, our substrate, it by itself or by supplying some energy, we'll see it in further detail later on, that how this leaving group is made to quickly leave and form this carbocation and the reaction is initiated. So this was how Sn1 proceeds. So if we see what is the rate of the reaction, it actually depends only on the substrate, only on alkyl halide, or it may, if our leaving group is halogen, if it is any X group, so it depends on our substrate. So this is how the rate of Sn1 substitution reactions depends only on the substrate. So that's why these are called Sn substitution 1 reactions. Similarly, we have Sn2 reactions. In Sn2 reactions, substitution but of second order. In such reactions, there is no formation of carbocation. Here in these reactions, if we have a substrate R1, R2, R3 and any leaving group X, nucleophile, it attacks on our saturated system and leaving group leaves. So these both steps take place simultaneously. How this happens is that if we see in the term of the orbitals, if we have this CX bond, which is carbon and leaving group, its antibonding orbital is attacked upon by nucleophile. Nucleophile has non-bonding electrons. So there is shift of electrons from non-bonding electrons to the antibonding sigma orbital of the CX bond. Okay, and in this step, transition state is formed. transition state is formed. Transition state is our substrate forms simultaneously two bonds. Nucleophile starts to form bond with substrate, means partially bonding of the nucleophile with the substrate, and partially the bond with the leaving group breaks. So this is our transition state forms. In Sn1, intermediate was formed. Here transition state is formed. Difference is that transition state is not isolated. We can't isolate the transition state. Transition state forms for very small time. It has a time period of much small that we can't isolate it, and this is highly unstable. Means if you try to trap it, it changes its paths, its energy increases or decreases. Hence transition state cannot be isolated. So here if we see substitution of second order Sn2, this proceeds through the attack of nucleophile on substrate and simultaneously leaving of the leaving group. Hence in this reaction there is involvement of both nucleophile and substrate in the rate determining step, means in the first step, which is the slowest step. Here this is only one step reaction in which nucleophile and our substrate both are involved. So rate of the reaction that is going to depend on substrate as well as leaving group, on nucleophile. So hence this is a second order reaction. This is a second order reaction. So if it involves nucleophile also in the first step, means it is initiated by attack of the nucleophile. So in Sn2 reaction we need a very strong nucleophile. So there are various other conditions that are to be discussed in detail, like what type of leaving groups favors Sn1, Sn2, and all these we will study in the preceding videos in the detail about Sn1 and Sn2 mechanism as well. Thank you.